So welcome guys, and thank you for coming to my presentation today. I will be giving a lecture on the SARS-CoV-2 spike protein. So if you're not aware, SARS-CoV-2 is the virus that causes COVID-19, which is a pretty relevant topic in this day and age. So first off, I'm going to be introducing the spike protein, also known as the S protein. So the S protein mediates viral entry. It allows the COVID-19 virus, SARS-CoV-2, to enter the cell. And the way it does this is by binding to this yellow protein down here called ACE2, which is to ACE2, which is this yellow protein down here. Let's see. There we go. Because S protein is so prominently displayed on the coronavirus, because it's like on the surface of the coronavirus, it's also a very common target for vaccines and antibody therapies. And in addition, the S protein has two parts. So you have the head here, and then the stalk here, which is kind of funny, right? Because it doesn't actually look that much like a spike. In fact, to me, it looks more like a chicken drumstick. So brief detour here. Before I talk more about the S protein, you guys should know about cryo-electron microscopy. So cryo-EM, as it's also known, is a really, really powerful technique for being able to visualize and see the structures of proteins and other really big molecules. So in the case of COVID-19, this background here that you can see with the colored coronavirus is actually an image that it's actually one of the first images of SARS-CoV-2. It was created using cryo-electron microscopy at a research lab in Beijing. And the way that cryo-EM works is it takes a bunch of 2D slices of a 2D slices rather of a 3D object. So you can see down here with the vase. So you can see down here with the vase that if you take 2D slices of it, you get a bunch of circles. But if you put that together, you can actually reconstruct what the 3D object looks like. It's actually a little bit more complicated than this, but this gets at the basic gist of it. And the thing about cryo-EM that is really cool is that it gives you atomic level resolution. So you can actually pinpoint the exact place that each individual atom is, which I think is really cool. Another thing about cryo-EM that's really awesome is that it's very cutting edge, and it's much easier. It's a much easier technique to find structures than previous methods, such as X-ray crystallography. So X-ray crystallography is a method where you have to try to form crystals of proteins, and then you fire X-rays through them. And then that way you can determine the structure based on like diffraction patterns and stuff. It's actually the technique that was used to figure out the structure of DNA, right? With Rosalind Franklin and all of that. So it's a pretty old method. But it used to be like the most common method for visualizing proteins until cryo-EM came along and sort of like took its place. One funny anecdote actually. There's a lab I used to work at in San Diego back in the summer before my sophomore year, and my mentor, she sometimes made this joke that like she had done X-ray crystallography before for proteins, and once she joked that like for X-ray crystallography you have to look at the phases of the moon because the crystallization is that sensitive. It's very, very difficult to do and can take months or even years to get it right. So that's cryo-EM. So today's presentation will be based on a paper I read in Science magazine. I actually found it on Twitter, actually, which is the top image. It's the tweet I found this from, and it's this paper that's used cryo-EM actually to determine the structure of the S protein, of SARS-CoV-2 S protein, at an extremely high resolution, and found out some really interesting things about it. And I'm also going to be taking a lot of images from the New York Times article called "The Coronavirus Unveiled." It's pretty cool. I recommend you guys check out these two articles later. The link is in this presentation, actually, and I'll be sharing the presentation with you guys later, both in the Zoom chat and over Slack. So the first takeaway from this paper that I read is that S actually has 3 flexible hinges, which is quite amazing, I think. So because these are scientists, they gave them extremely creative names. First, you have the hip, and then you have the knee, and then the ankle at the base. Let me turn on the annotation so I can draw on this. Okay, cool. So you have the hip up here, which, and then the knee, and then the ankle. You can also see this in this cryo-EM graph. You can see that like these blue arrows, they refer to the upper leg, as the scientists called it. So that goes from the knee to the head, And then these orange arrows demarcate where the lower part of the leg is. So that's from the ankle to the knee. I'm sorry, this should be hip. So as you can tell here from these angle numbers, these have a really, really high amount of freedom to like move around. In fact, in the paper, they describe some of the S proteins that they found on SARS-CoV-2 as being bent over like 90 degrees, which gives you a good idea of how like flexible these things are. And the really cool thing about these flexible hinges is that they're unprecedented. Like no other protein that is like S has these kinds of features. Other viruses, they do have like these kinds of what are known as viral fusion proteins, basically proteins that jet out on the surface of the virus and allow it to enter cells. But no other protein has these flexible hinges. Influenza has one, but that only goes to like 25 degrees or so. But this one has like 3 hinges, and each of them is almost like 20 degrees or more of freedom. Now, the thing about these hinges is that they're hypothesized to be able to help with viral entry, right? Because like these viruses are really tiny compared to like the ginormous human cells they're trying to get into. So essentially the surface of the cell is like flat to these tiny little viruses. So being able to have their S proteins move around with such great freedom would probably help a lot with viral entry. The other thing about the S protein being super flexible is that it could interfere with antibody activity. So antibodies are immune system proteins. They're basically like heat-seeking missiles, but they don't explode. What they do is they latch on to the proteins of these viruses, and they either target them so that other immune system cells can come around and clean them up, or they prevent the proteins from working at all. So in the case of the S protein, the antibody might block it so that it can't even latch on to ACE2 in the first place. But since S is so flexible, it may help with keeping off these antibodies. Okay, so part of the reason why the researchers were able to figure out that S has 3 hinges is because they ran what's known as a molecular dynamics simulation. So this is pretty cool. The gist of it is that once they knew the position of every atom, right? Remember, this is cryo-electron microscopy, so they have incredibly high resolution of the protein itself. And so basically what they did is they simulate every single atom, and then they run it to see how it'll move around. So they created this incredible video that I want to show you guys here. So you can see here that like the S protein is really, really flexible, right? You can see it moving around. I mean, let me actually leave this. You can see it moving around at both the hip, the knee, and the ankle joints. And also, you guys might notice that there are these little green things that are hanging on the S protein. This is not an error. This is something really cool, and it's actually the next thing I want to get to. Oops, sorry. So the second big thing about S, from the paper I got, is that it's actually protected by what's basically a sugar shield. So these big blue things that kind of look like clumps of like moss or something clinging to the S protein, that's actually sugar. So this is what's technically known as N-glycosylation, N-linked glycosylation, where N means nitrogen. So basically you have short chains of monosaccharides that actually bind to the nitrogen atoms of the amino acids inside the S protein. Now the cool thing about the sugar shield is that it basically like hides the S protein from antibodies, because antibodies, they are very, very specific to a specific protein. They can recognize a very specific region of our protein. But if you have like basically sugar, right? Sugar blocking these regions, and it makes it very hard for antibodies to figure out like what they should latch onto. Oops, sorry, there we go. Okay, so yeah, one last thing. If you guys remember the drawing at the very beginning of the S protein, so you might be wondering like how the S protein itself is still able to latch on to ACE2, as you can see down here, the yellow protein, even though it has all these clumps of sugar all around it. And it's actually kind of funny. There's this little light blue piece of the protein here that's like a hook. It actually like swivels up when the virus is near a cell in order to be able to hook onto ACE2, which I think is pretty cool. So that's all I got from the paper. So thank you guys for listening to my presentation. And if you guys want to read more about this, I highly encourage you to read more about this. These are the links to the articles and to the article in the paper I read. So make sure to check it out later. Again, I'll post the slide link in the Zoom chat and also in Slack. So thank you guys for listening.